Hello, everyone. My name is Samia Mahmood. I am an undergraduate student at Missouri State. My research advisor was Dr. Gary Mines in the Department of Chemistry and Biochemistry. My research project was identification of sugar-base proton interaction using NOESY two-dimensional NMR in Dickerson dodecamer, T9, Atheno C9, and 8-MAR-T for GPG sequences. The goal here was to understand DNA from a structural perspective so that we can see how conformational changes occurs when there is a damaged base pair in a specific sequence of DNA, so that we can use that knowledge to our advantage to understand how these specific damage sites get recognized during DNA repair. So to start with some background, I'm going to zoom in here. We have our four DNA nucleotide bases, adenine, guanine, cytosine, and thymine. These are marked here as proton 8 or proton 6. This is the number prime system that we use to complete a full NOESY walk. We also have our deoxyribose sugar, which is also marked as 1', 2', 2 double prime, 3', and 4' hydrogen. These are also the proton of interest that we investigate in our experiment. So to begin with the Dickerson dodecamer sequence, my data is right here. On the left, I have my 1' walk, and on the right, I have my 2 double prime walk. This is a very well-known, well-studied sequence. We use the sequence to our advantage to learn how NOESY two-dimensional NMR works and how to complete a successful walk. So to walk you through a little bit of the walk, here we have our cytosine 1 base pair, which interacted with the guanine 2 base pair. Then guanine 2 base pair interacted with the guanine 2 1' hydrogen. Then guanine 2 1' hydrogen interacted with the cytosine 3 base pair. Cytosine 3 base pair interacted with the cytosine 3 1' hydrogen. So the way it works is base interacts with the 1' proton, and then 1' proton interacts with the next base. And this is how we complete a full successful walk of NOESY. So using the knowledge that we gained from the Dickerson dodecamer, we ran another sequence called T9. This was also used as training. This sequence was very similar to the Dickerson dodecamer sequence, except we had mismatched thymine at the 9th position instead of the cytosine base pair that we had in the Dickerson dodecamer sequence. And here are my data of that. We also tried to run another sequence called Atheno C9. Unfortunately, we couldn't get this sequence in the right pH range of 7.35 to 7.45. And when we got it anywhere closer to that range and ran our NMR, we unfortunately didn't get any intense peak to complete full data inquiry and gather enough data to continue. Right here, we have our 8-MAR-T for GPG sequence. So on the left, I have my 1' walk. In the middle, I have my 2 double prime walk. And then on the right, I have my 4' hydrogen assignments. So we use our 4' hydrogen assignments to find proton-proton interaction with the phosphate group of the DNA backbone. We do this by using the HSQC spectrum. This hasn't been done yet for this specific sequence. This is something we need to do in future. And for the Atheno C9 sequence, if we want to continue with this sequence, then we would have to get more DNA samples so we can have better data to process. Thank you everyone for listening to my presentation.